Good day, everyone, and thanks for taking time from your busy schedules to allow me to share with you work that's been ongoing in my lab for several years. And what I'm going to talk about in particular is bioanalytical tools that we have been developing for disease diagnostics. And I will talk specifically today about cancer and our COVID-19 work. But before I get into the technical talk, I just wanted to say a couple words about Ralph Adams, who this award is named after. Rolf spent his entire career at the University of Kansas after receiving his PhD from Princeton in 1953. And in 55, he joined the faculty. And Rolf was known for a variety of things, in particular, neuroscience and using electrochemistry for studying neurotransmitters. His first PhD student was actually Ted Kawana, and Ted, in and of himself, is known for a variety of things, in particular spectroelectrochemistry. Unfortunately, Ted died several months ago, so I just wanted to bring his name forward and the fact that he was my PhD advisor. And another influential person in my life is my wife, and this wonderful person has allowed me to do all this work for so many years and has been very supportive. So what am I going to talk about today? I'm going to talk about liquid biopsy markers because you can develop the best tools in the world. If you don't have the right markers to enable those tools, they're not going to amount to much. And then I'm going to talk about microfluidics, in particular plastic microfluidics, because that is the technology platform that we'll be using for managing and talking about the disease diagnostics that I'll discuss with you today. And I'll have two examples specifically, namely using nanoscale vesicles or extracellular vesicles for subtyping breast cancer patients, using circulating tumor cells for managing pancreatic cancer diseases, and then an instrument for detecting COVID-19 using SARS-CoV-2 particles enriched from saliva samples, then finish up with a few conclusions. And then one thing I wanted to state was that our work and the work that I'll be talking about today has been founded a lot and supported a lot by this Center for Biomodular Multiscale Systems for Precision Medicine. This is a biotechnology center funded by NIH, and our mission is basically to disseminate new technologies into the biomedical community. All right, first off, what are liquid biopsy markers and why are they so important? Well, let's compare and contrast those to solid tissue biopsies, which are typically the standard of care for managing many cancer-related diseases. And the challenges with these solid tissue biopsies is that they're an invasive process to secure them. You're getting solid tissue from a particular organ. The other thing that you need to remember is that solid tissue biopsies are managing and looking at the primary tumor and not necessarily the metastatic sites. And the biology at the metastatic sites, especially for metastatic disease, can be quite different than what's going on at the primary tumor. The other problem with this is it limits the frequency of sampling, and sometimes based upon anatomical considerations, it's hard to get a tissue biopsy. And that's particularly true in the case of the pancreas. Well, liquid biopsies obviate a lot of those issues. Typically, they're less invasive. There's no surgery. Less invasive to minimally invasive because it's drawn from a simple blood sample. It samples both the primary and the metastatic sites, and it allows for frequent testing so the oncologist can respond faster to therapeutic changes by a particular patient. And it can be used for both solid tumors and liquid tumors or leukemia. So what are these liquid biopsy markers? The first one are the circulating tumor cells, and those are shown here. And I'll talk about this shortly. But the challenge with these is that they're very low in abundancy in blood samples, and typically 1 to 100 CTCs per mil of blood, and up to 10 billion red blood cells. So they're a major analytical challenge in order to isolate those markers from blood samples. The next one is the cell-free DNA, or the circulating tumor DNA. And these are small pieces of DNA that have been generated from cells undergoing necrosis or apoptosis. And they can be quite frequent or abundant in blood samples. And the challenge that you need to remember, though, is that you're particularly interested in the DNA that's carrying mutations. Those are the ones that we're looking at in particular, and those can be very low in abundance. In many cases, especially for early stage disease, about 2.8 molecules per mil. And the other one that I'll just mention here is the extracellular vesicles, also known as small EVs or exosomes. These are about 30 to 150 nanometers in diameter, and they typically contain nucleic acids, proteins, and other material. And even their abundancy can be quite high, even for early stage disease. But the challenge is the number of molecules or the disease-associated EVs in that 1 millisample can be very low. And that puts challenge on the number of molecules that you actually can use for a particular follow-up assay to manage the particular patient. So these are the markers that are typically classified as liquid biopsy markers. In particular, today I'll talk about circulating tumor cells and the exosomes. All right, so any diagnostic platform has to keep in mind that there's certain criteria that must be imposed upon it, in particular for in vitro diagnostics. And the most important thing that I can tell you is that after one measurement, the technology or the platform has to be discarded. It has to be disposable. And it has some profound implications on the production of these microfluidic platforms for in vitro diagnostics. And there's some particularly attractive attributes associated with using microfluidics for in vitro diagnostics. And some of them I've italicized here: reduced processing time, process automation, closed architecture. And that's particularly attractive because it allows you to minimize any contamination issues that can give you false positive results. But be careful. Challenges with a lot of the microfluidics is that there has to be a production pipeline to keep in line with the need for disposable devices to accommodate the in vitro diagnostics. And that's always a challenge. And other things, fabrication is always a very challenging endeavor for microfluidics because of the complexity of these platforms and the fact that you have to be careful with the surface chemistry. And I'll be talking about that today too. All right, so what is your recourse then in developing microfluidic for in vitro diagnostics? And our choice here, and this is kind of our niche, is to use plastic-based microfluidics. And there's a reason for that. And the reason is that it's very conducive to high-scale production. So you can produce lots of devices at in a very high throughput fashion and at low cost. And that really fits the mold for in vitro diagnostics. And a lot of this is predicated on replication-based technologies. For example, injection molding. And you can see this injection molding machine running here. The nice thing about injection molding, as the number of pieces or units required goes up, the cost goes down precipitously. And that's unlike PDMS, which with a production mode that's basically independent of scaling in terms of the number of units required for any measurement. So this is what makes plastics pretty attractive, is that there's production lines in place to do high scale production at low cost. And these injection molding machines were typically used for CDD, Blu-ray, and DVD disc production, and now they're being repurposed for microfluidic applications. All right, so what about the surface chemistry of plastics? And what you're looking at here is a variety of plastics: PMMA, COC, cyclic olefin copolymer, and polycarbonate. Well, it just so happened, the first thing you'll notice is that the surface chemistry is completely different for all of these plastics, not too surprisingly. But let's take a look at polymethyl methacrylate, also known as plexiglass. If I take that material and expose it to UV ozone light or O2 plasma, it undergoes a photooxidation reaction and generates carboxylic acid groups from this methyl ester. And that has several consequences. Number one is it changes the wettability of that. So this is indicative, this is the water contact angle, and this is indicative of a very hydrophobic surface that has a 70-degree contact angle. And when you expose it to UV ozone light, that contact angle goes down. It becomes more wettable. The other thing is you'll notice that you're generating these carboxylic acid groups here. And what that allows you to do is to generate a functional scaffold from which you can attach primary amine-containing entities directly onto that surface. The other unique thing about plastics is that after following this activation process with either one of these sources, they do not show hydrophobic recovery, as does with the case with polydimethyl siloxane, PDMS. And we've characterized these surfaces quite extensively after this activation process by using X-ray photoelectron spectroscopy to look at carbon-oxygen ratios. And you can see that those go up precipitously upon activation for all three of these polymer materials that I'm showing here. And their wettabilities after activation is almost comparable to that associated with glass. All right, so now we have a foundational basis for microfluidics and using plastics as the substrate of choice for manufacturing a microfluidic platform for a diagnostic application. So the first thing I'm going to be talking about is breast cancer and molecular subtyping breast cancer. And one of the things you have to remember is that breast cancer is a term that covers a lot of different diseases. And in fact, the standard of care typically involves once an individual, a female, has been diagnosed with breast cancer, they look at the receptor status of certain hormones to decide what is the recourse for treatment. And the treatment regimen is highly predicated on the receptor status. And typically that is done by using what's called this Prosigna PAM50 assay. And basically what happens here is about 50 or so genes are interrogated in terms of their expression by looking at the messenger RNA that's transcribed from a particular gene. And then patients are classified into 4 different groups: luminal A and B, HER2 enriched, and basal-like, which is also called the triple-negative breast cancer. And you're looking at the percentages of people that have these particular subtypes. And you can look from this heat map that's generated from all these genes that are shown here. There are about 50 genes here. And you'll notice that the pattern is distinctly different for each subtype. And that's basically what the molecular oncologist will look at, is to look at the mRNA expression profiling for these genes. All right, so, oh yes, and if you look at the Kaplan-Meier plot, you'll clearly see differences in the survival for different breast cancer patients with these different subtypes. Triple negative has a much lower survival rate than does the luminal A subtype. So how is this expression profiling done? And right now, the standard of care uses what's called this Prosigna nanostring assay. And basically what that involves is taking the messenger RNA out after it's been harvested out of formalin-fixed paraffin-embedded tissue, a solid tissue biopsy. It's incubated with a reporter probe and a capture probe that has biotin on its 3' end. And this is through a Watson-Crick base pairing that recognizes these particular sequences that are unique to this messenger RNA target. Also on the reporter probe is fluorescent dyes, the order of which allows you to deduce the identity of the messenger RNA. So after those are allowed to be incubated and form these complexes, the biotin is associated with streptavidin on these streptavidin-coated slides. They're put in an electric field, stretched out, and then you do a fluorescence image of these single messenger RNA molecules, and the reporter probes to decipher what messenger RNA is there. And you actually count them one by one and build up the expression profile. This requires about 100 nanograms of solid tissue in order to do. So what our job was in this particular case then was to use extracellular vesicles or exosomes as a replacement for the solid tissue biopsy. And this is just giving you a little bit more information on these exosomes or EVs, small EVs. They're relatively small. You can see that here. The amount of nucleic acids that's contained within these can be quite small, about 10,000 nucleotides. So even though the abundance of these, the numbers of these EVs, especially the disease-associated ones, may be high, the total amount of genetic material can be low because of their small size. And these are generated from all cells in the body fluids, not just the cancer-associated cells. So these are typically isolated, for example, using ultracentrifugation or precipitation methods. And these basically get all of the EVs, irrespective of the source, the cellular source of these EVs. And that does create a problem. And you can see that in this small little study that was done by Shannon Stott and her group in 2015. Several genes were expression profiled. Genes from the pure exosomes, these are the disease exosomes, and those from the ultracentrifugation. And you can see the expression profile for these genes is markedly different. And then if you use an affinity selection that uses a disease-associated marker, you can see the expression profile from this fraction is a little bit more similar to that of the pure exosomes, the disease-associated exosomes. And what you learn here is that affinity selection of the EVs that are disease associated is paramount to get really robust molecular information. All right, so what is the microchip that we used for this particular assay? This is actually the chip. This is done with our commercial partner, Biofluidica. This is a chip that measures about 38 by 42 millimeters in size. It's made from a plastic, in particular cyclic olefin polymer, via injection molding. It consists of 1.5 million pillars that are about 10 by 10 microns in size and 50 microns in height with about a 50 micron spacing. So why was it engineered with this particular architecture? And there are several concise reasons for that. Number one, the interpost spacing is small to minimize diffusional distances because the antibodies are chemically attached to the wall of these pillars. And you'll see how that attachment goes in just a few minutes here. And that small interpost spacing reduces diffusional distances. And then the other thing, the number of pillars is high in order to increase the dynamic range. So if we want quantitative information, we need to have a fairly large dynamic range in terms of the load of the EVs that can be surface packed onto all of these pillars. And you can see, even at relatively high volume flow rates, we still get really good recovery because of this small interpost spacing. The other thing, the length of these beds was engineered in order to maximize recovery because the EVs need to travel a certain distance down this array of pillars in order to interact with the antibody that's poised on the surface of these pillars. Then these antibodies are attached onto this surface using what we call a photocleavable heterobifunctional linker. So you can see the antibody right here. We take this activated plastic with these carboxylic acid groups, and then we add this bifunctional linker through EDC NHS coupling chemistry. This linker contains a coumarin moiety, and what we can do is shine blue light on that, and within 2 minutes, we quantitatively release all of the captured EVs from the surface of this chip so we can subject it to downstream molecular processing. And we've developed the infrastructure to do high throughput processing and managing the operation of these fluidic chips by using liquid handling robots. All right, so what do we need to detect? What are the EVs that we want to earmark? for this. And what we've done here is showing you what happens when cancer cells or tumor cells undergo what's called this epithelial to mesenchymal transition. These epithelial cells go into what's called a mesenchymal phenotype. And when they do that, certain proteins are upregulated, certain proteins are downregulated. And what we've done in order to accommodate that, we've cast a bigger net in order to get both the mesenchymal and the epithelial cells, because they can give you different information on the state of the cancer. The epithelial marker is called EpCAM, and it's generated from this cell line. And the mesenchymal cells are using fibroblast activation protein alpha, which is a serine protease, and it's found primarily in the invading front of cells, in particular the invatopodia, and it's found in 90% of all cells. And what you'll see here in this pattern, staining pattern for these two cell types is that the HS578T has high expression of vimentin, while the SKBR3 does not, and vice versa for the EpCAM. Vimentin is a marker of EMT, so these are more indicative of mesenchymal cells, and these are indicative of epithelial cells. All right, so we ran a clinical study where we collected both Fab alpha and EpCAM bearing EVs using the chip that I showed you on the previous slide. And we expression profiled those using genes that I'm showing you down here. There's about 25 genes that we used here. And this is showing you the expression pattern for different samples that were run. And what we were able to do then is to expression profile these EVs, both the EpCAM and the Fab alpha EVs, and then discern the molecular subtype of that breast cancer patient. Luminal A, HER2, luminal B, and basal. And what we found is that to correctly identify the basal subtype, we used the EpCAM EVs, and the Fab alpha EVs were used for all other subtypes. And when we did that, the call accuracy, the correct call for the subtype, as compared to the formalin fixed paraffin embedded tissue, was 100%. Okay, so now, what about CTCs? We looked in particular at circulating tumor cells for pancreatic cancer. And this is a terrible disease because the number of cases of pancreatic cancer is growing tremendously. It's the fourth deadliest cancer right now, and that's going to become even worse in the years to come. And the 5-year survival rate is very poor, less than 5%. And typically the reason for that is it's diagnosed at a late stage. So what we were interested in doing is helping to generate new therapies for pancreatic cancer using CTCs as a marker for response to therapy. And I'm going to show you that right now. This is really needed because there has been very few new treatment regimens for people with pancreatic cancer over the last 20 years. All right, so what are CTCs? And I'm going to show you this short video so you can get a handle on the importance of circulating tumor cells as a liquid biopsy marker.
When cancer spreads, tumor cells detach from the primary tumor and enter the bloodstream and travel to different parts of the body, a process known as metastasis. For patients with advanced cancer, the presence of CTCs in the bloodstream is associated with poor prognosis. These circulating tumor cells are rare and until now have been technically very difficult to detect.
So as the authors stated, the analytical challenge is that they're extremely rare, and there's billions of red blood cells and millions of white blood cells that you don't want to gather up. So you specifically want the tumor cells. So what have we done here? So what we've done is we've generated a different microfluidic chip, again made from a plastic, that consists of an array of sinusoidal channels that are typically about 25 microns in width and about 150 microns in depth. And in this array, there's about 250 channels. And the engineering was purposely done in order to provide high recovery. And you'll see why that manifests itself shortly. high purity because we don't want to have a high amount of white blood cells that carry over into the molecular assay. And this is done by putting antibodies on the surface of these sinusoidal channels. So this will process 2 mils of blood in under 40 minutes. And what that means is about 8 million cells per second that it will process. And what you're looking at here is blood, whole blood, that's running through this chip. And then after a buffer wash, you see no signs of non-specific absorption artifacts. All right, so why are those sinusoids placed into this chip? And oh yeah, let me say one more thing too, that typically, because we're using a top-down strategy to manufacture these chips, we have to put a cover plate on them. And we do that by using what's called thermal fusion bonding. And that basically means we put a cover plate of the same plastic over the substrate that has the appropriate microstructures in it with controlled temperature and pressure. And that allows the polymer chains in the cover plate and the substrate to intermingle to get really tight seals. The process yield rate on making these chips is well over 95%. All right, so the sinusoids. So when cells roll through the sinusoid, there's centrifugal forces that act upon them and they push them towards the wall where the antibodies are located. When they do that, it increases the contact time of the cell with the antibody. And because there's a flow rate that's acting on this hydrodynamic flow that's pushing the cells in the longitudinal direction down the length of these channels, they tend to roll along this wall, and that gives them more opportunities to interact with the antibodies that are stationary and anchored to this wall. And the advantage of that, you can see right here, this is a chip with pillars in it. What happens with the pillar chip is the interaction time is much shorter than the sinusoids that we've engineered here. And the advantage of that is we can operate at a higher velocity, higher throughput, and we actually get higher recoveries with that particular type of feature engineered into the microfluidic. All right, so what's the application here? So what we did is we set out with our clinical collaborator to look at niraparib as a possible therapeutic for pancreatic cancer patients. And PARP is poly-ADP ribose polymerase, which is a group of proteins that actually repair single-strand DNA damage. And what happens is the inhibitors are there to sequester the activity of that PARP inhibitor. That allows you more efficient killing of the cancer cells by using niraparib. And niraparib is actually an FDA-approved drug for ovarian cancer, and this was approved in 2016. And now we're looking for repurposing that drug for pancreatic cancer. And oh yes, one other thing is that BRCA mutations must be implicit in the tumor in order to be eligible for this particular therapy. All right, so what you're looking at here is, again, we're using anti-EpCAM and anti-Fab alpha antibodies to actually gather up both of these different types of CTCs. And then we're plotting the ratio of CTC Fab over CTC EpCAM as a function of treatment cycle. Each treatment cycle is for 29 days. And the first thing you'll notice is that in this particular patient, this ratio is getting smaller. That means either the EpCAM or the number of Fab CTCs is decreasing at a higher rate than the CTC EpCAM. And the other thing you'll notice, if we correlate that with the CT data, you can notice here that the tumor is indeed getting smaller. So this is a patient that's responding to therapy. What about one that is not responding to therapy? And in this particular case, this ratio is getting larger. And when it gets larger and you look at the CT scans, you can indeed see that the tumor volume, primary tumor volume, is getting larger. So this looks very promising as a liquid biopsy marker to actually deduce response to therapy, in particular niraparib. And what you're looking at here is looking at this Phi ratio, which is CTC Fab over CTC EpCAM, for progressive disease versus stable disease. And there's a statistically significant difference, and it's more robust than CA19-9, which is a serum protein marker that can be used for monitoring response to therapy. And what's very interesting, if you look at the Kaplan-Meier plots for progression-free survival, those that show always a decreasing ratio, and this is shown delta phi, which is the phi ratio for one stage of therapy versus the previous stage. This number is always getting smaller. These patients typically have a longer progression-free survival than those that do not show a decreasing phi ratio. All right, so the last thing that I wanted to share with you then today was our work in COVID-19, an at-home test using microfluidics as the foundational base for at-home testing for COVID-19. And as you all know, PCR and antigen antibody tests are kind of a hallmark for testing for COVID-19, but they're fraught with challenges. And you'll see some of those as I move along. I kind of summarize them here. What we are doing is a particle-based measurement. We're looking for active SARS-CoV-2 virus particles in a clinical sample and using the number of particles as an indicator of COVID-19 infection. And what's particularly interesting, and I just have to say this to start with, what's particularly interesting about this assay is that we are looking for active viral particles. That means the patients that have COVID-19 are infectious. And in many cases, the results that you secure from PCR and antigen antibody tests do not give you that information. And that's kind of discerned in this particular plot here. So what you'll notice is that this is showing you the onset of disease, and then when PCR tests and antibody tests are actually allowing you to get positive results. And you can see that these results can linger for days and weeks after the initial infection, when infectivity drops precipitously. And the reason for this is that because in many cases, viral particles will be shedding RNA into circulation very late in the stage of the disease, even after infectivity. And it's being done so from both active and non-active viral particles. The other thing that you need to remember, just to summarize real quickly here, is that antibody, antigen tests, and PCR tests require reagents, primers, enzymes for the PCR, labeled nucleotides, lots of reagents. And these have actually limited the onset of testing when the pandemic started in the first place. We had to ramp up production. All right, so what are we going to do here? We're looking for SARS-CoV-2 particles in saliva. We're going to use that as our sample of choice. So I'm not getting off my liquid biopsy march. We're still using the SARS-CoV-2 as a liquid biopsy marker, but now we're using saliva as the input sample. And we're basically using the same exact chip that I showed you earlier for the exosomes. So there's no change here. The only difference is that the readout is going to be different. We're not going to use this nano string assay for readout. But here's what's particularly nice is that this microfluidic assay that we'll show is fully automated and can provide results in less than 20 minutes. And it's using a reagent-free approach to secure those particular results. All right, so we take the viral particle or the exosome selection chip that has these 1.5 million pillars. We use this photocleavable linker here, this heterofunctional photocleavable linker, and we attach onto that a recognition element. And in this particular case, the recognition element is an aptamer. It's a short oligonucleotide of 51 nucleotides that binds to the receptor binding domain of the spike protein of SARS-CoV-2. Now think about that for a minute. you must have an available or vacant receptor binding domain of the spike protein in order for us to get a positive result in the assay. Otherwise, the aptamer is not going to latch on to the SARS-CoV-2 particle. And you have to keep in mind is that neutralizing antibodies, either from immunization or naturally occurring, will attack that receptor binding domain that binds to ACE2 receptors in the lining of your lung. All right, so the photocleavable linker is particularly attractive. In 2 minutes, we can release any particle that's been captured up and gathered up by this aptamer. So how does this work in terms of the selection? And what I'm showing you here is the specificity results. You can see that there's other human coronaviruses endogenously in your body, and we do not gather those up. Our aptamer does not bind to those human coronaviruses, nor does it bind to another type of virus, HRSV in this particular case. However, if you look at the active SARS-CoV-2, our recovery is about 94%, which is very attractive. High recovery, meaning we can look at very low loads, viral loads, and still get positive result. But if you use a non-specific aptamer, it goes away. What about the specificity in terms of non-specific absorption? That is, do we bind any free RNA? And after we run through a sample that's seeded with SARS-CoV-2, and then process that, and then release it and look at the wash through, the effluent, you can see that there's no positive results from after the photocleavage because of the fact that we have high specificity in terms of not gathering up the free RNA. All right, so what about the readout? Well, here we're using a label-free readout strategy. And it's basically a nano culture counter. And this is very old technology. And basically what this consists of is two reservoirs that have between them a nanopore or a nanoconstriction. When we bias this pore, put a voltage across it, particles move through here and they interrupt the flux of ions moving through these pores. And we can register that as a response, as you can see here. And we can size particles based upon the amplitude of the signal that's generated. The challenge is that these are not very concentration dependent. The limited detection for concentration is not so impressive, about greater than 1 million particles per mil. And that's going to be a problem because the range of SARS-CoV-2 in saliva samples can run from 10 to the third to 10 to the 8th particles per mil. So this is showing you schematically of a particle moving through a constriction right here that's biased with an electric field applied across it. And you can see the light here that's generated is indicative of the perturbation of the ion flux through that constriction when this particle is present. And not too surprisingly, the signal that you generate, the amplitude of the signal that you generate is highly dependent upon the particle size and the size of the pore, this pore right here that's engineered into the readout strategy. And people have done work on the utility of using resistive pulse sensing compared to existing technologies for sizing and counting particles. And you can see it compares very favorably with standard of the art technologies for example, transmission electron microscopy and NTA sensing for reading particles. So here is the chip that we engineered to address specifically the concentration limited detection. We have a constriction, a pore, that's about 350 nanometers in depth and width because the range of particle sizes for SARS-CoV-2 is about 150 nanometers in terms of the average. The range goes from about 80 nanometers all the way up to about 250 nanometers or so. And the chip has 5 pores in parallel and with the proper fluidics to give you high sampling efficiency. So instead of getting a sampling efficiency of 10 to the 10 to the minus fourth percent, we get sampling efficiencies on the order of 2.7% using this particular configuration. And we validated those results using COMSOL simulations and doing particle tracking analysis. And you can see here's a chip that was made in a plastic. This is made in a plastic via injection molding. Okay, this is actually showing you data collected from that nanopore chip sensor. And what you can notice here, for a blank of PBS phosphate buffer saline background, we see no signals. But when we see that PBS with particles, we can clearly see these current transient spikes that have an amplitude of roughly about 155 picoamperes and a width of about 0.12 milliseconds. All right. So if you do look at the analytical figures of merit by doing a calibration plot, you can see that the calibration plot is linear over several orders of magnitude for particles with the number of false positive 0. We've done that by making sure the threshold detector is set at a level we get no false positives. We can process 30 to 50 microliter input volumes. Sampling efficiency, as I pointed out, is 2.7 here. And what that percent and what that amounts to is a limit of detection from of about 2.4 times 10 to the third particles per mil, but with the enrichment of the viral particle selection chip about 10 to 100 fold, that puts us in a limit of detection of about 24 to 240 particles per mil, and that makes it very comparable to PCR-based techniques. So what we did is we did a clinical study, and this is the last bit of data I'll show you. We did a clinical study with Sinusoids Diagnostics that does RT-qPCR for diagnosing COVID-19 from nasopharyngeal swabs and using RT-qPCR compared to our assay. And there's a lot of data on this slide, but I'm going to just, when we look at all negative samples, we agree 100% with the accepted protocol or the accept protocol for testing. But when we look at samples that are positive by the Sinusoids RT-qPCR, we can see in some cases where we concur with the positive results from RT-qPCR, but in half of them we do not. And what we notice is that the antibody concentration is much higher in these non-detected cases, indicating that these people have a non-accessible receptor binding domain of the spike protein, i.e., they're not infectious. So what we're doing right now is packaging this chip up into a single cartridge, and that's shown here. So here's the selection chip. This is this EV selection chip, and the VP selection chip has these 1.5 million pillars, the nano culture counter, and we've actually manufactured this already. This is made from all plastics. These chips with the proper connections sit onto a fluidic motherboard with the fluidic network, and the valving devices actually placed onto the motherboard. And we don't use PDMS for the valving membrane here. We're using elastomeric COC, and it has some unique properties that I won't go into in detail here, but just to point out that this can be injection molded, why PDMS, which is typically a valving layer for membrane valves, cannot. And we're packaging this all up into a handheld instrument, which is shown here. And we're in the process of testing this device right now. So what I was basically able to show you today in my talk was that liquid biopsies offer some unique opportunities for managing a variety of diseases, infectious diseases, cancer diseases, but also other diseases, cardiovascular diseases, stroke, infectious diseases. They have advantages and disadvantages. The most pressing disadvantage, or challenge I should say, is that they're mass limited. You have to keep in mind that you don't get nanograms of material. Sometimes it could be picograms to picograms of material. Microfluidics are particularly attractive for analyzing liquid biopsy samples, and they can process a variety of samples, blood, urine, saliva, and the like. And making microfluidics from plastics is particularly attractive for in vitro diagnostics. And finally, to keep in mind that we have mass limits whenever we use liquid biopsy markers, we have to think about new molecular approaches for analyzing their cargo. And we're in the process of developing those right now by showing the ability to do nanofabrication to make devices that have some exquisite opportunities in terms of looking at mass limited samples without requiring amplification strategies. So finally, I need to acknowledge my students who basically did all the work that I showed you today. That these people are tremendous people. They work very hard, even through the pandemic. And I wish I could talk more about them. They've done a tremendous job in terms of collecting up the results that I showed you today. Then we've been blessed with a lot of support from many agencies, institutes at the National Institutes of Health, in particular NIBIB, that supports our Biotechnology Resource Center. We have had commercial partners, Roche Diagnostics, Biofluidica, who's our partner in disseminating our technology in the commercial sector, and Stratec in terms of manufacturing. And then also 2 cancer centers that I've been associated with, the University of Kansas Cancer Center and the UNC Chapel Hill Lineberger Cancer Center, who's really helped us a lot with the collecting patient samples and thinking of new applications for our technology. So with that, I will stop and I will be happy to answer any questions that you might have. Thank you so much for your attention.